All right, so yeah, we'll move on to the second short talk of the day, which will be given by Dr. Jongyun Bae. So Dr. Bae is a postdoctoral fellow at the National Institute on Aging, and he will give a talk titled Validation of Neuroimaging Markers Using Omics. Oops, sorry, my bad. I didn't allow you to share your screen. I will do that now. Now you should be able to share your screen.
Sorry about that. Okay, let's get started. All right, good morning, everyone. My name is Jongyun Bae, and I'm a postdoctoral fellow at NIH. I changed the title and the content of the talk a little bit from the initial title to give more general overview of using omics data to validate the neuroimaging markers. So let's get started. Neuroimaging has been widely used in aging studies to infer changes in microstructures in aging as well as in AD disease progression. For instance, the left figure demonstrates different imaging measures such as Myelin Water Fraction and diffusion measures that reflects microstructure changes along aging as well as the advance in disease state. We can see the drastic reduction in myelination between the mid-age and the old subjects, and further loss of the myelination with the advance of the Alzheimer's disease. Also, similarly, the diffusion measures exhibits increased diffusivity in aged individuals, as well as in patients, suggesting the alteration in brain microstructures. Our lab has shown the correlation between these measures with different physiological changes, such as cognitive decline and motor function. My colleague will present this work more in detail this afternoon, so please stay tuned. But our study has shown that lower myelin content is associated with steeper declines in executive function, processing speed, verbal fluency, and memory. Also, another study from our lab has demonstrated that lower myelin content is also associated with lower and steeper decline in gait speed, suggesting its association with decline in motor function. Although these findings from neuroimaging has demonstrated strong association with physiological changes in aging and also in patients, direct validation of these measures are still necessary to confirm we are measuring what we claim to measure. When it comes to validation, most of us are appealed to perform histology, which can be very specific to finding what we desire. For instance, in one of the previous studies shown here in the left figure aimed to validate their Myelin Water Fraction measures with a histopathology from MS patients. You can see the MS lesions from the T2 weighted images and also from the hypointensity and the estimated Myelin Water Fraction maps. And this Myelin Water Fraction maps correlate well with the histology where myelin is stained with local fast blue. However, there are many limitations in using histology as a validation tool. First, the tissue sampling is quite invasive, and therefore performing the histology in large cohort covering all ages is not really practical. Second, the tissue preparation step induces several destructive aspects to the tissue, which can alter the integrity and the properties of the sample tissue. Lastly, the histology has limited coverage, which prohibits exploring the whole brain-wide validation of imaging markers. Therefore, we adopt different approach using omics to validate our neuroimaging measures. The omics datasets can provide more comprehensive understanding behind the molecular mechanism of the targeted tissues, by using, for instance, transcriptomics. Therefore, it may offer a whole brain-wide direct validation of imaging markers involved in the microstructural changes. So in this study, we adopted two omics data to validate two neuroimaging markers. And we used a proteomics to validate our neurite density index derived from diffusion imaging. And we used transcriptomics to validate our fluoximetry-based Myelin water fraction measures. So to measure the neurite density index, we used diffusion imaging to model different contributions from the intra-axonal water and the extra-axonal water. Our multi-component diffusion model assumes the intra-axonal water exhibits different diffusivity due to the restriction from the microstructures. Therefore, we can model this different contribution to the signal, which would serve as a proxy for the neurite density at each voxel. We adopted a NODI model, which considers three different compartments: intracellular with more restricted water, less restricted water in extracellular, and free water in the CSF with isotropic diffusion. However, with NODI, we found that inaccurate estimation of CSF water contribution causes the overestimation in NDI. As shown here on the NDI map, the high level of NDI is estimated in the ventricle, which is not physiologically reasonable. Therefore, we developed a constrained version of NODI called cNODI, where we adopted the fraction of CSF water from FSL toolbox and constrains the optimization for the rest of the parameters. As shown here in the bottom right, now we see the NDI values that are more plausible to the physiological values. Using this, we obtained NDI measures from 59 cognitively unimpaired subjects with age spanning from 21 to 81 years old. For the validation of our NDI measures, we collected blood sample and obtained neurofilament light chain levels in the plasma. So NFL is one of the 4 subunits of the neurofilaments, and it has been shown to indicate the axonal damage and neural cell death. And this can be sampled from both CSF and blood. We collected samples from 43 subjects among the study cohort mentioned earlier. And our hypothesis here is that the level of NFL will be negatively correlated with our NDI measures. Next, we aim to validate our Myelin water fraction measures. We developed a fluoximetry-based Myelin water fraction method called Bayesian Monte Carlo multi-component derived despot, or BMC-MEC despot. The water trapped in myelin is suggested to experience distinctively shorter T2 relaxation rate as compared to the intra or extra-axonal water. Therefore, we can compute the fraction of the water contributing from the myelin, which reflects the level of the cerebral myelination. BMC-MEC despot consists of 2 imaging sequences, spoiler echo and BSSFP. Although I'm not going to discuss all the details about the signal modeling here, but we adopted the Bayesian approach to solve the high dimensional estimation problem to measure Myelin water fraction, T1 and T2 of the 2 compartments. We measured the whole brain Myelin water fraction from total of 132 subjects with age between 22 and 94. And these subjects are without any known impairment in their cognitive state. To validate our Myelin water fraction measures, we use transcriptomics data from Allen Human Brain Atlas datasets. And this HBA dataset offers microarray measures to infer whole brain-wide gene expression. The dataset includes more than 20,000 genes that were detected from 3,700 tissue samples from 6 donor brains. You can kind of see the visual representation of the location of tissue samples across 6 donors in this figure. We use Abidjan Toolbox for processing gene expression and use different atlas to compute the regional gene expression for all genes in several white matter regions as some deep gray matter structures. So our hypothesis here is that Myelin water fraction will positively correlate with gene expression specifically related to myelin. So here are the results. First, we see the NDI measures across aging process in different brain regions, shown here on the left figure. And we observe this inverted U shape of NDI trend for most of the brain regions, which suggests neuroid maturation and subsequent loss with aging. On the right, we see a strong and significant correlation between NFL levels in plasma and NDI measures in different lobes of the brain for each subject. So this strong correlation suggests that our NDI measures directly reflects the axonal damages. And for the validation of Myelin water fraction measurements, although this is preliminary at the moment, we found strong and significant correlation with genes that are related to myelin. For instance, one of the genes here, myelin basic protein, is a protein found in the cytoplasmic space in the bilayer membranes of myelin sheath. We can see the white matter regions exhibiting high myelin water fraction demonstrates high gene expression for this protein, while deep gray matter structures with low myelin fraction shows relatively lower gene expression. Other than this gene, we also found several transcription factors that are related to the transcription of myelin, which correlates well with our myelin water fraction measurements. In summary, we have validated two neuroimaging markers with omics datasets. We found NDI measures from our diffusion imaging negatively correlated with plasma NFL level, which is known for axonal degeneration. We then used transcriptomics to validate our myelin water fraction measurements, where we found strong correlation between myelin water fraction and the gene expression that are related to myelin and the transcription of myelin. However, there are several limitations of the current studies. First, obviously, unlike proteomic measures, the myelin water fraction was measured from different subjects than those from the transcriptomics datasets. However, we aim to simply demonstrate the feasibility of using different omics study to validate the neuroimaging findings here. Also, the plasma level of NFL concentration can be substantially lower compared to that in CSF. Therefore, we are looking into correlating the NDI measures with NFL levels from CSF samples. Lastly, it will be interesting also to see whether the correlation with gene expression varies with age. So those donors from the microarray datasets, they have different age, different spectrum of age. So we can we can vary this, we can stratify by age and hopefully gain more information about the molecular mechanism behind the cerebral myelination with aging. With that, I'd like to thank our lab members and also thank you for your attention. I'll be happy to answer any questions.
All right. Thank you so much, Jongyun. That was a great talk. Chris, any questions in the chat?
Yes, thanks for a great talk. Did you happen to see any correlation between naughty and myelin water fraction parameters?
In specific to naughty, so not C-naughty, just naughty. Well, I mean, if you can refer to this paper and it actually compares the naughty NDI measures from naughty and the C-naughty, and I believe also ODI as well. But yes, we saw a similar correlation between the NDI measures with the myelin water fraction. Oh, sorry, the myelin water fraction. That's a good question. So I'd have to get back to you on that. I'd have to look through I don't think we did in this particular paper. I don't think we did the compare with Myelin Water Fraction, but I think we are working on those correlation between the diffusion measures with the Myelin Water Fraction as well.
Awesome. Mustafa Buhara mentioned that they did, and they gave me a link, so I will share that in the chat for those interested. Christine Tardif was wondering, for myelin water imaging, did you repeat this analysis with other MRI metrics, such as proton density, which we know also correlates with myelin content, but isn't myelin content? Just sort of wondering if this correlation with gene expression is as strong, or maybe it's muted when you use a different MRI metric.
That's a very interesting point. So as I said, we are still at the very early stage of this transcriptomic analysis, but that will be another interesting area that we want to investigate into, such as relaxation rates, T1, T2 with the gene expression, or any other measures, neuroimaging metrics that we can correlate to with the gene expression. But for our intuitive thought at the beginning was to relate this Myelin Water Fraction, which is the direct proxy of the myelination, to the gene expression that is related to myelin.
Awesome, thank you. Maybe in the interest of time, one more question from Jean Chen. So with a lot of diffusion modeling, you make some assumptions. She was wondering if your model assumptions are still valid when myelin is damaged, like in multiple sclerosis.
Right. That's an interesting point. I don't have an answer right as of right now, but that's an interesting point. What to test whether our assumption or the constraint that we impose are still valid in the different state of the brain microstructures. But yes, so we'll definitely look into those as well.